Hey guys, what's up? It's AP Bio day 14 in the third 9 weeks, and we are doing lots in the way of genetics labs. So, Friday, you guys did a gel electrophoresis lab. It was on the HNPCC gene, and I posted a video for this. Here's our gel chamber, or a mock-up of what the gel chamber looks in the different individuals. So, you guys in here, you in the gel, you noticed there were how many bands? Anybody remember?
13.
Okay, so there are 13 different individuals, and so when the DNA spread out, like how many different bands did y'all see?
2 or 3.
Anybody remember?
There were several.
Where's your picture? Y'all get out your pictures. Y'all forgotten everything. There was either one or the other.
It's been a while.
It's been a while. It's been since Friday a while.
Yeah.
What happened in that time?
What? No, it's not a long time. So in the lab, remember, we were studying a genetic disorder that's directly related to colon cancer. And so how do we know which individual's in there? Now, keep in mind, really, we're only focused on numbers 5 through 13 because these are the actual people that we looked at their DNA. The others were either for comparison, what it looks like if you had it, or what the control looks like, and this is for determining how big the DNA fragments are. So how many bands did the DNA break up into for the individuals?
Looks like 3.
Okay, they either had 3 or they had what?
1.
Thank you. So, the one, okay, so normally you've got this trait, you got you get one gene from mom, you get one gene from dad. And so wherever this is on the chromosome that they're cutting this, why did some of them end up with 3 bands and some of them ended up with just 1? Who can tell me? The 1 wasn't actually 1, it was 2, but they were where? They were stacked. They were on top of one another. So if you see one band like that, it doesn't necessarily mean it's one band. It could be like 2 of those bands on top of one another. So you had one from mom, one from dad. They had the normal gene for this caretaker gene, and so typically doesn't show up as or cause colon cancer. Now, in the individuals that had 3 bands, why did they have 3 bands?
Their strand was longer. They had the gene.
It was longer. The
Smaller.
farther it runs, what does that tell you about the DNA? Smaller it is. So what happened, Miranda?
They have the gene for it?
Yeah, so that tells us they had the gene, and so the restriction enzyme that was used cut at a certain place on that gene where it identifies that gene. And so basically what happens is, so you would have like a chromosome and then it identifies that gene and it cuts it. And when it cuts it, it splits it in 2. Okay? So if you've got 1 in the normal place, that means like your normal one from maybe like mom, and then maybe you got the one from dad that coded for cancer, and so because it was cut by the restriction enzyme, it split it into 2 segments of DNA. Does that make sense? So any of the ones that you see that have 3 different segments, those guys are going to have that autosomal dominant trait that is likely to cause that type of colon cancer. Okay? Any questions? So, I want you guys to hold on to this. Y'all started making a pedigree the other day. I think it's on 282 in your book maybe, where it talks about pedigrees. We are not really ready for pedigrees yet, and so I want to wait until we get to pedigrees to finish that part up. But you guys can go ahead and go down through here and mark which ones have that gene that is going to be dominant for causing that type of colon cancer. Okay? Any questions? All right, next thing. Let's look at and talk about your lab today. We need to go ahead and set this joker up and get it ready. So you guys today, if you'll flip in your lab packets there's one page that looks like this, and then with a bunch of circles on it, and then there's another page that looks like this. So this is Transformation Kit Quick Lab This is for the pglo lab that we get from Biorad, this company out of California. And this quick lab is what you're going to be actually using tomorrow. So, the gist of the lab. Here we go. In the lab, you're going to have some bacteria. So today we're going to have some E. coli. It's in a little test tube, and it's in the fridge behind me. And so you are going to take that bacteria, and in order for transformation to happen where we actually get the bacteria to take up some DNA and get them transformed, in this case they're taking up that gene where they'll glow, you guys need to actually grow some bacteria first. So what we're going to do today is you're going to need a petri dish, and you're going to get some bacteria, and we are going to streak our starter plates. And streak it just means rubbing the bacteria all over the outside of the plate. Okay? So that's what we're going to do today. Once you're done with that, then you're going to put them in the prep room. I've got an incubator back there that is set at 37 degrees Celsius, which is the same temperature as what? 37 degrees Celsius is 98.6 degrees Fahrenheit, which is, body temperature. So why are we setting the E. coli to that temperature to grow?
Because that's where we normally grow.
Yeah, that's the temperature they normally grow and where they do the best. So that's what we're working on today. So you're going to put bacteria on your starter plate. We've already got the starter plates made. You need to have your gloves and your goggles on. And then I'll show you guys at home. I'll write in some clips or paste in a clip of this. And so you guys at home can kind of see what we're doing in terms of how we're actually growing these bacteria on the starter plates. And then tomorrow when you get in here, we're going to have to work very quickly because of the time we have. And we're going to go through the quick guide today a little bit later on. But we're going to go through there and we're going to complete the transformation lab. So you guys are going to have to be in here and get started and work very, very quickly in order to get that done in one lab period. And hopefully the next day you guys will come in and look at the incubators and we'll shine the UV light on them, and those bacteria, if transformation took place, they will be glowing. Yeah, so they have a gene for fluorescence from a jellyfish, and that's in a plasmid, and so if those bacteria take up that plasmid, then they will glow too. Okay? Any questions? So, this is what you're doing today. So, I'm going to have, I'm going to get a petri dish out for you. So, here's our petri dish. Okay? Be kind, be confident. Remember that. So, on your petri dish, you are going to have hold on a second. Okay, so here's what's going to happen. You got this inoculating loop here, this little thing. You'll see that at home, what we're using? There we go. Now, there's a little circle on it. So you're going to come up, and there's going to be a test tube, and you're going to put this down in here, and I'm going to turn it kind of sideways. And so you're going to roll it around, and then you should get a little film on there, kind of like a bubble, like if you're blowing bubbles. Don't blow bubbles with this, though. So that's going to be bacteria that's on there. And then you're going to take that, you're going to grab a petri dish with a lid on it, and then it's going to look something like this, and you're going to run it along the top of your petri dish agar solution. So in here, you have a gel. It's an agar gel, which is derived from seaweed, I think. That's what they make it out of. It's kind of a gel, like a thick jello. And inside it, there is LB. LB is short for Loria broth. So think like chicken broth or beef broth or something like that you cook with. It's just something to provide nutrients for the bacteria. So you guys are going to take your inoculating loop. Now, I bent mine just a little bit here so that it would be kind of flat. Because if you have it straight and you dig it into, somebody last period or first period did this, they dug it into the agar, you're not going to get good bacterial colonies on there. So you want it to kind of just float, skim it barely on the top of the petri dish. You don't want it to dig down in the agar. It's not very thick, so it's easy to do. So you got to be careful. There's a fine line. You want to rub it on it, but you don't want to dig it down in there. So get it, put it down in there, and this is how you put it on. You want to streak it where you do one side, back and forth like this. Turn your petri dish, do it at like a diagonal, this way. Turn it again. Do your other diagonal. And then whatever my other diagonal is to be up and down. So you've turned it 4 times and you basically covered that dish with those bacteria that are on the inoculating loop. Does that make sense? So you're gonna get in your groups of 3. You're gonna get your petri dish. You're gonna come up and get your bacteria on the inoculating loop, and then go back, pop the top off, get it on there real good, put the top back on, and then write your initials on the underside of the petri dish, and we're gonna put it in the incubator in the back. And so overnight, we're just growing bacteria. Tomorrow when you get in here, if we grew some bacteria, if y'all didn't mess up or I didn't mess up, then we'll take those bacteria and we're gonna try to transform them tomorrow. Okay, does that make sense? Perfect.
Am I touching it? No.
Get it flatter on there.
Like this?
Flat. Back and forth. All the way across. Don't dig into it. Let it float. Let it glide. You're digging into it. Don't dig into it.
That's how not to do it.
That's exactly how not to do it. I mean, you only gotta get 1 or 2 good colonies, so. All right, now twist it. Oh, well, that one's better, except for that one mess up the end.
Yeah. Jinx.
Careful with this.
I agree, but y'all chose me to be a loser, so here we are.
That was their mistake.
Thing, you're not the leader.
We have a democracy. It's a very poor one.
Wow. I don't think I like that.
Gosh.
Now, let's open some of the flat parts here. We get some colonies growing by tomorrow, and you guys will see by then if we've actually grown any bacteria or if he's completely messed everything up. Hey guys, so we're going to talk about the transformation kit. Quick guide here real quick and what we're going to be doing tomorrow. For the people in class, it is your responsibility to look over this for homework because you will be performing the lab, and it's got some details in here you need to be aware of. So highlight, underline, write down questions if you have them. We have a very limited amount of time to do this tomorrow, so I want to go ahead and kind of give you a background of what's going to happen so you're not just coming in blind. You're basically going to take 2 of these little test tube looking thingies, okay? Or well, you're going to have a bunch of these actually, but we got 2 initially. And you are also going to have one of these little foam blocks here. The foam blocks actually, the foam pops out, and that's where you put your test tube, down in that hole. And so this is like your test tube holder for these. They go in there. And then you also have some pipettes that they'll be at your station for you to use, okay? For you guys at home in my class, then you're gonna see how this actually goes all down. Or the 3 of you who are doing this lab in another class and maybe you're remote and wondering how this works. So basically what's gonna happen here is you're gonna take your bacteria that you grew today, okay? So you already put them on the petri dish like we saw earlier with Mason and his group. Hopefully they start to grow. And tomorrow we're gonna pull them out of the incubator, and you should see little colonies. So the colonies are where an individual bacteria that you smeared on that petri dish started to grow and divide, okay? Remember, in the gel, there is LB in there. What do we say LB is? Lorry broth. So it's just basically a nutritive solution like a broth, like a chicken broth, beef broth, whatever, some kind of broth. And so it's a broth, it's a nutrient so the bacteria can grow and they'll divide and divide. And you wind up with little colonies that look like little bitty circles, okay? So they say to just get 1 of the colonies here. I don't really think it matters. Usually the more bacteria, the better, but however you want to go about doing this, depends on what your instructions say. But we're going to take some of those bacteria, make sure you get them on the end of your inoculating loop, the little yellow loop like we had a second ago. And you're basically going to take your bacteria, put it in one of those test tubes, put some of the plasmid in the test tube. You're going to cool it down, put it on ice. You're going to heat it up. You're going to cool it down. And during this process, you're going to expose the bacteria is going to kind of the membrane. Think of it like getting colder, getting hotter. It's going to expand and contract. And what you're trying to do is to allow that bacteria some flexibility to uptake some of that plasmid into it. Okay? So now you'll notice at the top of the page here, you see 4 petri dishes. So tomorrow you will have 4 petri dishes that you're doing this lab with. Okay? So you have one that says LB, which just has the Loria broth on it. The other one says LB and AMP. AMP is short for, feel free to write any of this in, ampicillin. Ampicillin is a type of?
Enzyme.
No. Ampicillin, what does it sound like?
Amplified.
Like penicillin, it is an antibiotic. What do antibiotics do?
Break down viruses.
Antibiotics kill? Bacteria. I'm glad y'all took this class. Y'all are learning a lot, I hope. All right, so antibiotics kill bacteria. What are we trying to grow on the plates? Bacteria. So what do you think is going to happen on that plate? Well, you would expect nothing, but if it has the plasmid, the bacteria has that plasmid has a gene for fluorescence or for glowing when exposed to UV light. It also has a gene on that plasmid somewhere. It has a gene for antibiotic ampicillin resistance. So what's going to happen is if it has that plasmid inside it, then that ampicillin isn't going to affect it. So it's going to be resistant to that ampicillin. Does that make sense? So that's another way to tell you, like, if something's growing on there, what's happened to it. Has it transformed or not? And then the last plate is going to be the LB AMP, and it's going to have ARA. ARA is short for arabinose. Arabinose is a type of? Sugar. It has OSE at the end, so you know it's a sugar. So that is another gene on that plasmid. So you have to have the arabinose in order for the pglo gene to be expressed. So it's one of those like tertiary genes that helps to coordinate or work with the pglo gene. Okay, so the arabinose has to be present in order for fluorescence to take place, because you got to have that to express that, for that gene to express and for it to start to glow. Okay, so that's basically what you're doing, and then you'll do your inoculating loops, and you'll put them on those 4 plates according to how it says and what it says to add there, whether the plasmid's present or not. You'll stack them up nice and neat, and you will put them in the incubator until the next day. We'll check them for 2 days after that to see if they've grown, but you should be able to see by Wednesday if your your bacteria have transformed and if they're glowing or not. They just continue to get bigger, the colonies, and they glow more and more. And what we'll do for extra credit is whichever group in here has the most bacterial transformation colonies, we'll give some extra credit. And if you don't have any, we'll probably have to count off. So be precise in your application and following of instructions. Okay. You will use this sheet right here. You see a bunch of circles and questions. This is where you're going to record like what happened on your Petri dishes, but that'll come along on Wednesday. And I think that's about it for that lab. So today we are going to talk about genes and organismal development. Got a few notes to go over. And you guys remind me on the lab packet that you have, Walker, y'all need to be on 376. On the lab packets that you have, we're going to do the parental analysis on there. Let's see if I've got it with me here. Now, on the lab you did Friday, you guys had to determine, here it is, you know, if the gene for that particular type of cancer was present or not. We're going to do a Maury Povich lab today where you're going to determine who's the baby's daddy, okay? So the lab looks something like this, and we'll work on that the last 5 or 10 minutes, okay? So let's look at genes and organismal development. Looks something like this. All right, Katie, read that first one for us. Go.
Genes are segments of DNA that are blueprints for making proteins and enzymes within the cells.
Okay. So the genes are the blueprints that determine what type of proteins and resulting enzymes, since most of those are made of protein, results from the cell. So you have certain coded DNA that's going to make those type of proteins or enzymes. Zygote, tell us about that one, Mariah.
One to N diploid cell that is a result of an N haploid sperm fertilizing a N haploid egg.
Okay, so let me clarify here. A zygote, when it says it's 2N, that means it has DNA from mom and it has DNA from dad. So that's where the 2N takes place. So you're getting we have 23 different chromosomes that code for different things. You get one set from mom, one set from dad, you wind up with 46, okay? We all started out as a zygote many, many years ago. If you want to know when you started out as a zygote, at about 9 months to your current age, and there's where you started out as your first cell. Yeah. Let's see here. And then all of the cells resulting from that should have the same genome or DNA, unless you have some what? What could change your DNA? Mutations. So all those cells should be the same unless there were some mutations that resulted. Which happens. I know sometimes I read a twin study where it said identical twins, they will have some mutations and some slight differences in their DNA. The next section is on cell differentiation or specialization. What does that mean, Tay?
Certain cells are made to do certain things.
Okay, so yeah, there are lots of different cell types in our body, and they have, all of them have different types of functions. So they are made a certain way to function a certain way, okay? And so you have a zygote that starts off, it starts dividing. All those cells are the same. What type of cells are they? Stem cells. Stem cells are said to be what? Meaning they can turn into any type of cell. Starts with a T. Totipotent. Totipotent means it has the total potential to become any type of cell in your body. Now, after the cell divides up to a certain point, it starts to develop into certain 3 different tissue layers. What are they called? What are those 3 early on different tissue layers that we develop into? You're close. Ectoderm, endoderm, mesoderm. So these are the 3 different layers that start to develop. Like the endoderm is primarily the lining of your digestive tract. Now, those cells can become many different cell types, like different parts of your digestive tract, but they can't become any type of cell. So they've already started to differentiate somewhat. So we call those cells, starts with a P. Pluripotent. I can tell none of y'all have read over the notes today because this is down a little bit further in the notes under stem cells. Thanks for preparing. All right, next thing, morphogenesis. Morph means the body shape. Genesis means the creation of. So how do we create the body shape that we have? Morphogenesis is the product of cell differentiation occurring during development. So early on, we all started out looking like what? A round ball of cells, right? Remember the morula, the round ball of cells, the whole develops in the middle, blastula. And then we all wind up being elongated, okay? We're taller than we are wide. So how does that go about happening? So there are genes that control that development. One of the genes that helps control development is a set of genes code for apoptosis. Apoptosis is when what happens? Okay, so apoptosis is programmed cell death. So meaning that your cells in some places are programmed to die. For example, where? Between your fingers and toes. Good. So that's how we form our fingers and toes. They first start out as webbed, and then the cells in here are programmed to die, and that's how we wind up with those appendages. Your tail. So all of us had a tail in utero, and it underwent programmed cell death, unless you wound up with the tail and it didn't work out that well. My aunt had a 3-inch long, she had like 3 extra inches on her tailbone. And when she was about 40, it gave her enough back problems, I think from sitting and stuff, that she had to have that 3 inches cut off her tailbone. So I also heard from my wife the other day, she knows somebody that had a tail. It's not like a wiggly tail, it's just usually like extra tailbone material.
Does she not have gotten
She could have. I don't know if it maybe didn't bother her then. They didn't. I'm sure they didn't have the money early on, and I'll have to ask her about that. She was in a medical journal because I don't know. It's not something that usually happens. So I'll see next time I see her if I can figure out what they thought caused it or why they waited so late. Now, apoptosis. So we talked about that, killing off the tail and between the fingers and toes. Plants also undergo a morphogenesis. Plant cells do not move as they are restricted by the cell wall. So they're pretty much solid, they're in place, they're adjoined to one another, okay? They mature in place and respond to the environment. So they can move, but we'll talk about how plants move a little bit later. It's not really like muscles like we have. It's actually cells changing shape. All right, number 2 under that, they continue to display growth until they die. The growth occurs at the apical meristem. Apical meristem is important. The apical meristem is the tissue on a plant at the end of the roots and the shoots. So like where the plant's going out on the end of the like limbs and at the roots where they're going out, okay? That tissue is very specialized because that's where new growth is taking place. So you have new cells developing there. Tomorrow, actually, we're going to be rooting some cells in the gardening club, rooting some fig trees. And we're going to basically be turning stem cells, not stem cells like we just talked about, cells on the stem of the plant, and turning that tissue, getting it to turn into root tissue. So we'll talk about that in the process of, getting that differentiation to occur. It's not that hard. So, morphogenesis in animals. Cells have moved into final position during birth, and animals display limited growth after that. So we get bigger, but your overall body shape and the way you look is pretty much done by the time you come out of your mama. All right, next thing, cloning and clones. What is a clone, Mariah?
100% genetically identical organisms.
Okay, where do we see clones in nature? Identical twins. Fraternal twins are not any more related than, any other brother or sister. But identical twins are 100% genetically identical, except for whatever few mutations may have taken place, like in utero. So sometimes it happens, but usually they're small mutations, and usually it's probably not something that you would see. All right, cloning. How do you get a clone? This is something you need to be able to articulate. Write out the different steps. So let's say, who wants to get cloned in here? Good, Mason, we want to clone himself. We can tell by his ego. So Mason, if you needed to clone yourself, what's the first thing we need?
Remove an egg cell from a female organism.
Okay, we need an egg cell from a female. So ladies, anybody want to donate an egg to him? Thanks, Katie. So Mason, what else do we need?
A haploid nucleus from an egg cell.
We don't need that yet. What else do we need for the ingredients?
Probably sperm.
No sperm necessary.
My DNA.
You need your DNA from where?
My chromosome.
Which ones? What kind of cell do we need DNA from?
Sex cell.
No sex cells. You're cloning someone. Sex cells are for diversity. What?
A somatic cell.
Yes, you need DNA. So we would need some DNA from one of Mason's somatic cells, one of his body cells that has 46 chromosomes. Now, since Katie loaned us an egg, what are we going to have to do with that egg? You almost said it, Morgan. What? So we've got to remove her DNA. We don't want her DNA in Mason's clone. That doesn't make any sense. So after we remove her DNA, what do we do?
Put them together.
How do you put them together? Do we put them together? What is needed in that egg now?
The somatic cell with the DNA.
You need the DNA from the body cell. Good. So you take the body cell's DNA from Mason and you put it in the egg, and now you have all of his same DNA as he has. And through some tricks of the trade, which I don't know, they say electrical or chemical or some kind of stimuli, they get that cell to start dividing. And those cells start dividing and divide and divide, and you wind up with a ball of cells. Now, there's 2 different types of cloning. What are they? You might know? Y'all watched the cloning video, didn't you?
Yeah.
You brains must have died since then. No brain altering drugs. There's two types of cloning, therapeutic and reproductive. Therapeutic is when you're cloning for,
Well, medical purposes.
yeah, tissues. So let's say Mason needs a kidney. In order to get that kidney, you're not going to grow a little baby Mason, knock it in the head with a hammer, and take a kidney out of it. Okay? That's not how therapeutic cloning works. We're going to grow that reproductive tissue that is identical to his DNA, and at some point before it starts developing into a little baby, we're going to get those cells and derive them into what? We're going to turn on some genetic switches, and you want those cells to turn on into kidney cells. And so you can grow kidney tissue that way. And then you can insert that kidney tissue. And then that's a way of getting more genetic material that you need, whatever it may be spinal tissue, like a spinal cord, like neurons. So that's therapeutic cloning. When you get to reproductive cloning, that's where like you take a Mason, you make a little baby Mason. In order to get to being a baby, what do we have to have? What do we have to do with that egg that you've putting his DNA in?
Put inside of them.
Yeah, it's got to grow in we got to find a uterus out there somewhere. So you got to put it in a uterus and it's got to start growing, and then would become a new little Mason. Now, are you going to have clones that are like the same age as one another, like you see in the movies? No, you can only clone someone and it grow up as a baby. So whatever age are, put in somebody, it's going to have to grow, it's going to have to start to develop. So you're going to have a little mini me, basically.
Can you have two clones that are the same age if you clone them at the same time?
Yeah, I mean, you could do that. You could you could take it and probably split that ball of cells, or you could clone them at the same time, and you could wind up with two little identical twins is basically what you'd wind up with.
Would they be like exactly identical?
They should be. It's the exact same DNA, right? Let's hope so.
Do you think that there could ever be the technology to do that? Not saying it would be.
Where you could have the same age person?
Yeah.
I don't know. They always show these like blank slate kind of people that they inject with cells or whatever. I just don't foresee that happening. I could be wrong, but I don't foresee it happening. All right, so we've already went through stem cells and talked about them in research. Now, stem cells, there's a lot of controversy because they're derived from what? Embryos. So what happened was they put a ban on the embryonic stem cells because of that, because basically you're taking little babies or early on that could develop into babies. So that was the controversy. And now they've actually discovered a way, scientists, to take skin cells, turn them back into stem cells, and use them for research. Less controversy, easier to do. You don't have to make little babies. Good to go. All right, pattern formation and positional formation. Both of these sets of genes just basically determine where things are located. Like if you're a fruit fly, where your wings going, where your legs going, that kind of thing. We'll talk more about those later on, but that's all the pattern and positional we're talking about. How the pattern of the organism is set up and where things go on that organism. Okay? So, we'll finish up genes and development tomorrow, and I want you guys to look at your lab packet under where it says, this is Megabucks, is the guy here. Okay. So we've got a Maury scenario. So this is your gel, okay? So like a gel that would go in the gel chamber, just like the other day. At the top, you have your wells where you put your DNA. So here's the scenario. Megabucks got a lot of money. X, Y, and Z are the baby's mamas. They all say that Megabucks is the dad, okay? And then you have their children. Megabucks doesn't want to pay child support on all those kids, especially if they're not his kids. So we're doing DNA tests to find out, are they his kids or not, okay? So what you're looking for is, circle all of the pieces of DNA that match up between the kids and just Megabucks.
Excuse this interruption. At the end of class, we need to see Ashlyn Kilgore.
She's not here.
Oh, okay, thank you.
Thank you. So, you want to see the ones that just match up with the kids and Megabucks. You may have some DNA that matches up with the kids. The mom and Megabucks, don't count that because it could have come from the mom. We want to know that ones that just match up with Megabucks, okay? So the easy way to do that is take a straight edge and put it along here, or you can even fold your paper up and do it like this, and you can look across there at the DNA and see which strands of DNA match up with him. Circle them, and out of those three, you should be able to tell which one or ones match up with him and which one is his kids. Okay? You guys take care.